our research investigated the effects of snake venom or potential effects of snake venom on cancer cells. More specifically, we looked at how snake venom might make those cancer cells break themselves down while leaving the normal cells unharmed. For our research, we broke it down into an introduction, kind of explaining why this research matters in the broader world. Then we broke it down to the methods that the researchers went by, and the results, the findings from those methods, and an interpretation, why those methods and results are important and the specific limitations of them, and we'll leave it with a conclusion.
So, let me begin with why this matters. The colorectal cancer, which is the cancer of the colon, is one of the leading causes of death worldwide. Even with today's best treatments like surgery, chemotherapy, radiation, many patients still face serious side effects, and sometimes the cancer resists treatment altogether. So, researchers are searching for new approaches, and one surprising path leads us to snake venom. The question guiding this research is simple. Can snake venom help push cancer cells toward death instead of survival? If yes, how does it do this? Understanding that process could help us design better therapies in the future. So let's look at some background concepts. Think of your cells like tiny factories. Cancer cells are factories that refuse to shut down, producing endlessly. Now these factories have two key systems, retirement and recycling. Retirement, called apoptosis, is when a cell shuts down on purpose. Recycling, called autophagy, is when a cell cleans house. Usually, recycling helps a cell survive, but if it goes into overdrive, the factory collapses. Snake venom contains a mix of proteins. Most of them are dangerous, but some of these proteins push cancer cells into recycling so much that they destroy themselves. This happens through a chain of signals inside the cell called the JNK pathway. You can picture it as a row of dominoes. Once they start falling, the message keeps going until the cells shut down. And interestingly, this domino chain falls more easily in cancer cells than in healthy ones. The idea behind this research is, what if venom can push cancer cells to recycle so much that they essentially break themselves down? So let's look at the methods part.
So what did the researchers actually do? They started with the colorectal cancer cells. These cancer cells were taken from the colon, and instead of being inside a person, they were grown in a dish in a lab. That way, the scientists could safely test different treatments and watch how the cells respond. The treatment they chose to test was cobra venom. They gave the cells different amounts or doses to see whether small, medium, or large amounts made a difference. Now, how did they measure these effects? They used three main checks. The first one was kind of a pulse check called MTT. You can think of this like checking whether the cells are alive and kicking or not. The second was a cell breakage test called LDH. If the cells are leaking, this test lights up, kind of like how a car's check engine light tells you something's wrong. The third check was what I would call a glow test under the microscope. They use a dye that makes recycling activity inside the cells glow. Normally, cells recycle a little to clean up waste, but if they recycle too much, it negatively impacts it. On top of these tests, they also looked at the signals inside the cells, which are instruction chains telling the cells what to do. They found that specifically the JNK pathway, which was once again like a row of dominoes falling, was turned on, and that chain reaction led the cells to die.
Moving on to the results section of the research, the researchers found that the snake venom made cancer cells recycle themselves too much until they broke down and died. And this happened through a signal chain pathway called the JNK pathway, which works like a switch that turns on cell death. Normal healthy cells were not harmed by the venom. So moving on from the bigger picture, let's look at the step-by-step of what the study showed in parts 1 through 5 and how snake venom triggers recycling in cancer cells and leads to their death. In part 1, the study showed cancer cells treated with snake venom glowing with more green dots. Those dots mean that the cells are recycling their insides too much. In normal cells, there was no change, which meant that the venom affected cancer cells, but not healthy ones. This told the researchers that the venom was targeting cancer cells, not normal ones. In the second part, the green dots got stronger over time, so the recycling kept increasing the longer the venom was there. When the researchers added another drug called HCQ, the dots became even brighter and more clustered. The bar graph backs this up, showing essentially that the venom raised recycling and venom plus HCQ raised it even more, which raised the question, if HCQ boosts recycling, what happens to cell death? In part 3, the study checked whether HCQ changed the venom's ability to kill cells. And so the bar graph shows more cancer cells survived and fewer died when HCQ was present. The blue stains pictures also show fewer dead cells with HCQ. This means that HCQ essentially blocked the venom from killing the cancer cells. In the fourth part, the researchers asked which path inside the cell the venom was using. So the usual growth path called mTOR stayed the same, and instead the J&K path turned on. So the bands were stronger, and when J&K was blocked, the green dots, which represent recycling, went down. So this essentially showed that the venom was working through the J&K pathway, not the normal mTOR pathway. And in the last part, when J&K was fully turned off, the venom could no longer kill the cancer cells. So the graphs show more cells survived and fewer died when J&K was blocked, and the pictures show fewer dying cells, which proves that the venom depends on the J&K switch to kill cancer cells. So taken all together, the study showed kind of a clear story that the snake venom pushes cancer cells to recycle themselves too much and die by flipping the J&K switch, and it kind of leaves the normal cells unharmed.
So what do these results actually tell us? The key idea is that cobra venom didn't just randomly kill all the cells. Instead, it set off a very specific chain of events inside the cells. The venom turned on the J&K pathway. Remember, that's a signal chain that's similar to dominoes falling we talked about earlier, and this pathway caused the cancer cells to recycle themselves way too much until they broke down. Importantly though, the venom didn't seem to hurt normal healthy cells. That means cobra venom could act as a selective weapon against cancer, targeting just the harmful cells while leaving the normal ones alone. This makes it stand out because many current cancer treatments, like chemotherapy, often damage healthy cells along with the cancer. This also shows the potential of natural therapies. Something as dangerous as snake venom, which we normally think of as poison, might actually be harnessed and reshaped into medicine. It's an example of the idea from poison to potential cure. But of course, there are limits. These tests were only done in dishes in a lab, not in people. Real venom is far too toxic to use directly, so scientists would need to find and isolate the safe parts of it. That means a lot more testing has to happen before this could ever become a real therapy. So in short, these results are exciting because they suggest venom could be developed into a natural-based cancer treatment, but we're still at the very early stages.
And that brings us to our conclusion. From this research, we have learned that poisons can become medicines, and nature is a great source of inspiration for science. It also shows that discovery can come from humility, as cures may come from unlikely places like snake venom. So for future direction, this research could isolate safe proteins, and researchers could move from testing in lab dishes to animals and eventually people to find applicable cancer treatments derived from snake venom. So there are several take-home messages from this research. One is that snake venom triggers cell death via over-recycling. Another take-home message is that this research is early, but it is very promising as it shows the power of nature in medical discovery. Additionally, this research teaches us hope, as healing can come from unexpected places like toxic snake venom. Thank you for your time.